Hello and welcome everyone to this video podcast for Biology 211's lab. This will be our first and our last podcast for Module 4, and in this module we're going to talk about Drosophila melanogaster. Let's go back to our plan for the week before we start talking about the fruit fly. We've already completed the first two parts for this week, labs 3.1, 3.2, though you still may be working on your poster. Remember that is due Friday. Lab 4.1 here, we're going to start working with Drosophila, as I said before, and we're going to spend a little time talking about Drosophila mutants, and there's going to be a little activity that you have to complete with the mutants. And then there is a genetic cross simulation that you will do using a program called Star Genetics. Don't forget your due dates shown down here. Everything is due either Friday or Saturday. The worksheets that we're going to be talking about today, they will be due on Saturday at 11:59 p.m. Again, here's just a visual of where we've been and where we're going. We're almost done with lab after this particular lab. We just have a couple things to do next week, including the poster evaluation. So as you have learned, there are many common model systems that geneticists use. We use bacteria, plants, yeast, worms, fruit flies, fish, mice, a lot of different organisms. In Biology 211, we have talked about many of these in the lab. We've talked about plants, yeast, worms, and now we're talking about fruit flies. Drosophila is often, as I just said, referred to as the common fruit fly, much like you might get flying around in your kitchen when you've left your bananas out too long. Drosophila has many features that make it a really excellent model organism. It's small, it's easy to grow in a lab, and it has a relatively short life cycle. It goes from egg to adult in about 2 weeks. It produces a large number of progeny. This is important, as we've talked about with other model organisms, because if we're going to use statistics to analyze our results, we need lots of progeny. Drosophila has been a staple amongst the model organisms for many years, and because of that, there are many hundreds of mutants available for people who study Drosophila. Like many other model organisms, Drosophila also has its genome completely sequenced. Of the organisms that we've talked about, it probably has the fewest number of chromosomes. But as we've talked about before, the number of chromosomes does not specify the complexity of an organism. So many people who are interested in studying human diseases will look at organisms like fruit flies. So as it says down here, 77% of human disease-associated genes share homology with fly genes. So we can learn a lot about a disease, a human disease, by first studying in an organism like fruit flies. Now it's going to be important that you go through this PowerPoint to do the first part of Lab 4.1. In Lab 4.1, you're going to need to describe wild type and mutants, and so you're going to need the next several slides to help you do that. And as I said, you're going to use this information to complete the first 5 questions on worksheet 4.1, and it's worth a total of 10 points. So the first question on this worksheet is that you need to describe the phenotype of a wild type fly. And then you're going to need to describe how each of 8 different mutants are different than wild type, and that's question 2. And then you will put your skills to the test and ID various fly mutants. And the fly ID quiz is located in this PowerPoint. And in that quiz, you'll identify the unknown fly mutants with one of those 8 mutants. You'll see in a moment, and I'll show them to you, that flies with subtle phenotypes will be presented with a comparison to wild type. The unknown will be circled in green. But first, let's take a quick peek at the mutants and wild type. So first, this is just showing you the difference between males and females. The easiest way to tell the difference between a male and female fruit fly, at least for me, is to look at the abdomen. In males, it's more rounded and it's darker, as you can see down here. Females, the abdomen is pointed and it's lighter, as you can see down here. Males do have sex combs present on their front legs, and females do not. But for me, it's always quicker just to look at their abdomen. So wild type flies, they have a very distinctive red eye. Though this is just a cartoon, you can see that the wings are nicely formed and have a nice symmetry to them. The body color is not real dark, it's more of a tan or brown color, and antennae are present. So let's look at the mutants here. The first mutant is called vestigial, and you can see vestigial has abnormal wing phenotypes. You can see they look like they're shriveled up here, like it was left outside for too long. And obviously, as you could probably guess, these flies do not fly. Apterous, its main identifying phenotype is that it lacks wings. And as I'm sure it will be no surprise to you, these flies do not fly either. Now the next 2 mutants we're going to look at are based on the color of the abdomen. The first one we're going to look at is yellow. So you can see the color of the wild type abdomen here and the color of the yellow mutant here. You can see that wild type is a little darker, and it's a little easier to see here, where you see this more yellow tinted abdomen compared to this more darker and a little cream colored here abdomen of the wild type. And the next one we're going to look at is ebony. So here's ebony. Wild type flies here, and then the ebony mutants here. These are a little bit more distinguishable than the yellow abdomen phenotype. Their abdomens are a much darker color, and the contrast is a little stronger. So it's easier to identify those, I think, than the yellow. White is also a pretty easy mutant to identify because instead of this big old red eye, they have this big old white eye. So sepia here is also an eye color mutation. Wild type, of course, is this big old red eye, and sepia has this darker eye, almost like a very dark red color. Bar is a very interesting mutation. You can see in wild type, we've seen that very large eye. Bar has variable phenotypes, but it consists of this very narrow eye, and it's caused by a gene duplication like we talked about in lecture. And the more duplications there are, the more narrow the eye becomes. Antennapedia. This might be the most interesting one you'll see here. And so in wild type, you see over here, you see the antenna. But in antennapedia, where the antenna should be, they actually have legs growing out. So no antenna, and they've been replaced with legs. Now what you're going to want to do on your own is take the Fly ID quiz. Using your notes from questions 1 and 2, identify the unknown fly mutants on the next 2 slides. The Fly ID quiz is question 3 on worksheet 4.1. For each of the 8 unknowns, select which mutant is shown. Answers will be one of the 8 mutants: vestigial, apterous, yellow, ebony, white, sepia, bar, or antennapedia. And they won't be repeated. Just one mutant is used, and after you've used it, it won't be used again. I mentioned before, if it's a subtle phenotype, there will be a picture of wild type next to it. And I already mentioned this, but mutants will only be used once. Mark your answers on worksheet 4.1, and you will upload that to the Blackboard shell when you turn in this worksheet. So I'm not going to go over these. These are things you're going to have to go over on your own, but you can see on this slide there are 4 mutants. And here you'll see sometimes we've indicated what the wild type is, and in doing so we put a red with a mark through it, and the mutant that we want you to identify here is in the green. Okay, same for these 2 mutants here. After completing the Drosophila quiz, there are 2 final questions. 4th question asks you to do a Google search for interesting fly mutant names. Drosophila researchers have a wide range in flexibility on what they can call their mutants. And due to that, over the years, they have come up with some very interesting and entertaining mutant names. And if you just Google interesting fruit fly mutant names, you're going to see what I'm talking about. A lot of these interesting names will pop up. You'll probably come up with a site that says something like 15 most interesting Drosophila mutant names or something like that. I want you to just list 2 of those that you find interesting. And then the 5th question asks you, what is a potential danger in naming fruit fly mutants these fun, interesting, entertaining names? Keeping in mind that many genes in fruit flies are linked to human diseases. All right, we're going to move to part 2 of Lab 4.1. And in this, we're going to do what we would call an in silico exercise, where we're going to do a genetic cross, but it's going to be a simulation of a genetic cross. Now, experiments or research conducted or produced by means of a computer modeling system, we call these in silico. These are very useful in learning these concepts and understanding how they work because they don't use live animals. This is all done with computer-generated animals. You'll see in this system we're going to use that you will specify the genetic background of flies to be used, and the program will provide the outcome for you. You don't have to wait for 2 weeks for the fly to develop. This is an excellent way to practice some of your quantitative reasoning skills, to generate and test hypotheses about modes of inheritance. And this also comes at a very good timing because in lecture we're also covering this, and this will help you get ready for the exam coming up. We're going to use, as I said, this system called Star Genetics. And there are a few things that we've covered in lecture that will be helpful if you review, or at least have available to you as you begin these Star Genetic crosses. One of those is using the chi-square goodness of fit test. In class, we just call this the chi-square test. Now, in class, we also talked about lethal alleles. I'm not going to go over this slide because we've done this in class. This is also a figure from your textbook. The main thing I want you to remember with a lethal allele is that when you take 2 heterozygotes for a mutation that you're interested in and you cross them, if it's a lethal allele, you should see a 2-to-1 phenotypic ratio. And the reason for that, remember, is that one of the homozygotes, in this case big Y, dies in utero. And so it's not counted as part of the progeny because we never see it. But with the yellow mouse experiments here, 2 of the mice are yellow and 1 is non-yellow. But again, look for that 2-to-1 ratio. That is a key to identifying a lethal allele. You'll also want to review complementation that we talked about in lecture. The main thing I want you to remember here is if you take 2 mutants and you cross them together, and if the progeny appear as wild type, then we say that those 2 mutants complemented each other. In this case, mutant 1 here provided a non-mutated copy of gene 2. Mutant 2 here provided a non-mutated copy of gene 1. So the progeny have a complement of non-mutated genes 1 and 2. Now if you take 2 mutants and you cross them together and the progeny appear as the original mutants, in this case no wings produces no wings, we say they do not complement each other. Because in this case each mutant, mutant 1 and 2, provided a mutated copy of gene 1. So the progeny, while the gene 2 is perfectly fine, gene 1 is mutated because both parents provided a mutated copy. So they do not complement each other. When this lab is taught face-to-face, we still use the in silico because we know it's a very efficient way to do these fly crosses. In doing this, you will need a free software program called Star Genetics. You have to complete the worksheets on your own and then upload them to the lab Blackboard shell when you turn in your worksheets for this module. There are 2 ways to access Star Genetics software. The first way, and this is the way I would not initially recommend it, but you can try it this way, is to download it directly to your computer. The reason sometimes this doesn't work is depending upon your computer, you may have to update your security for Java. And if you know how to do that, then that works great. If you don't, then that might cause problems. But you can give it a try. Here's the website right there. This is a different way to do it, and I think this will be less problematic. You can use CMU Citrix environment, and what you'll do is you'll go to this website and you'll follow these instructions. What I wanted to do though is go through this with you because the first time I did this, it wasn't as clear as I had thought these instructions were. So let me go to this website here and then let's follow these instructions. Okay, so here's that website, and then when you enter that website here, it'll take you to the Citrix storefront. Just put in your username with your password. It's the same password you use when you log into any of your CMISH accounts. Now once here, the instructions tell you to go to desktops, so I'm going to click on desktops. And then it tells you to go over here to Biochem, EAS, Physics, and CPS. So that's what I'll do. It asks if I want to open the Citrix Workspace Launcher, and I say yes. And then it opens up this new window for us. And it's set up more on a PC landscape here. And when I first logged onto this, I did not know how to get anywhere until I realized I have to hit this little window down here, which you probably all knew. It was just me. And then over here it tells you to go to the Star Genetics. And it's over here on the left, and you just go by alphabetical order till you get to Star Genetics. And then you click on Star Genetics. And it'll take you right to the program. And then down here it tells you how to get started. So you click on File, and then you click on New. And then over here are a whole bunch of exercises you can do. You only need to do 2 of them. You can do them all after the class is over and you're bored and you're wanting to do more genetics in the middle of July or August. And then you can come back and do these. But for now, you need to do Fruit Fly Exercise 1, Level 1. And then you will need to do Fruit Fly Exercise 1, Level 2. So I'm going to click on the first one for you. Now, as you're going through the Star Genetics, go to your lab worksheets, and it's going to give you a step-by-step introduction in how to use, and it's going to give you a step-by-step instructions on how to do the crosses. The first few questions here you can see are just definitions. So it begins here by telling you that you're going to start a research project in a genetics lab that studies fruit flies. Your advisor decides to give you an interesting mutant called grounded that has just been discovered. In addition, you are also given 2 true breeding homozygous wild-type male and female flies. All 3 flies can be found within the strains box. And so what is the strains box? Let's look at that here next. So we go back over to Star Genetics. This is our strain box here. You have your grounded and you have your 2 wild-type. And you can see your grounded mutant is female because it gives you the little symbol for female. Then I have some questions here for you to answer. The first is, what is true breeding? You can look that up. The wild type fly is true breeding, but you don't know if the grounded mutation is recessive or dominant. Therefore, what are all the possible genotypes? So you'll need to think about all the possible genotypes that this grounded mutant is. Alright, I'm going to skip down here to question 3. You want to determine the genotype of grounded. You are unsure whether its phenotype is caused by dominant or recessive allele. You are also not sure if grounded is true breeding or not. To determine its genotype, cross-grounded with one of the wild type flies. And so let's go ahead and do that. So we're going to come over here to our strains box, and we're going to put it over here into the mating site. And since this is female, we need to pick a male wild type. So we're going to pick this guy here, and we're going to put him over here. And then we're going to mate them by just hitting mate. And now this tells us all of the progeny we want. Now in some of the experiments it might tell you to mate again, add more matings, but for now we're just going to stick with this. You can always start over by hitting discard, or you can do a new experiment and it'll keep both sets of experiments. You can also sort them, so here they are sorted. The first several here are all the mutant, and then the last ones are all the wild type. You can look at the summary of them. It tells you 48% of them are mutant and 52% of them are wild type. And it breaks it down by male and female as well. Okay, let's go back to our sheet. Here it tells you to go ahead, after you do the cross, to indicate how many flies are wild type, how many are grounded, and so forth. And then it will ask you what is the ratio of those mutants. And then it'll ask you if they're dominant or recessive with an explanation. Now I'm not going to go through each of these questions, but you get the general idea of how you're going to take flies, you're going to mate them together, and then you're going to look at the results. And the instructions here are really well laid out, step by step. If you run into any problems at all, let myself know or let Rodney know, and we might be able to address it through an email, or we might be able to pop onto Teams and we can pull up your cross simulations and look at them together. Now you see how the Star Genetics works. As I mentioned before, you'll need to complete Exercise 1, Level 1, and Exercise 1, Level 2. Together, these are worth 60 points. You can work on this with your teammates. In fact, I strongly encourage you that you do work on this with your teammates. But everyone has to submit their own completed worksheets. As I said, make sure you review chi-square, lethal alleles, and complementations. These will be important in one or both of the exercises. I didn't list it here, but you also need to be familiar with just the general concepts of Mendelian and non-Mendelian inheritance, as we've talked about in lecture at this time. All right, looking ahead to next week, our last week, we're going to have poster evaluations, and then there's going to be something I'm going to do to replace the debates. I'll give you a little bit more details on this in lecture as the debates, but hopefully it'll be useful for us all. All right, let me know or let your TA know if you have any questions, but if not, we'll see you in class or in lab. Bye for now.